Hi, everybody. My name is Haley Brazier. I'm the Donald M. Kerr Curator of Natural History here at the High Desert Museum. Welcome to our Natural History Pub Series. We obviously planned this to originally be in person, but due to the really high COVID numbers here in the county, we've decided to move it online. So I appreciate everyone's flexibility as we make these sort of changes as need be to keep everyone as safe as possible. I'd like to thank the Pacific Northwest Forest Association, also known as Old Smokies, for supporting this event. And the High Desert Museum sits in and shares the stories of what was, is, and always will be the Indigenous Plateau. We extend our respect and gratitude to the Confederated Tribes of Warm Springs and all of the Native communities who have lived here for thousands of generations and continue to thrive here today. So before I introduce my speaker, I'd like to mention that our next Natural History Pub will take place on Monday, February 7th, 2022, from 7 to 8 p.m. We're hoping that COVID numbers are better by then and we can hold that event in person at McMinimans. We'll be hearing from state biologists Corey Heath and Andrew Walsh on mule deer. So if you'd like to learn more about that event or RSVP, please head over to the High Desert Museum website and navigate to the February calendar where you can look at the page and RSVP for that event. So now I feel very honored to introduce our speaker for tonight, Jodi Wilmoth. Jodi is the Native Fish Society Steward for the Little Deschutes River and the Oregon representative for the Trout Unlimited National Leadership Council. She has a degree in molecular biology and has been an avid fly fisherman for 20 years. Conservation of native species has always been very important to her. Most recently, since moving to Central Oregon, she has been involved in Oregon spotted frog and native fish conservation and participated in Trout Unlimited's youth education program. Tonight, Jodi is presenting research that was gathered on the traditional homelands of the Klamath, Modoc, and Yahuskin tribes. So Jodi is going to speak for about 45 minutes, and then at the end of the presentation, there'll be about 10 to 15 minutes of time for her to respond to your questions. If you have questions, feel free at any moment to just go ahead and put them down into the chat box, and I will be the MC at the end. So also we are recording this presentation, and so you can go up onto the YouTube website of the High Desert Museum in a few days after this presentation to watch it there. So without further ado, I'll turn it over to Jodi. Thank you.
So hi, everybody. Again, my name is Jodi Wilmoth, and I am a Native Fish Society Steward for the Little Deschutes River. I'm really excited to be here tonight to be talking to you about this project. I'm really excited about this project and some of the implications that it can have for bull trout as well as other endangered species, and how this technology can be used throughout the basin and throughout the West to establish habitats for critical or for endangered species, threatened species. So we'll get into exactly what this technology is and how we're using it here in the Deschutes Basin. So the first thing that I want to do, I know that most people, when they give a presentation, they usually save their thanks until last, but I want to actually put this at the very beginning of my presentation, because I wouldn't be here tonight unless it was for these people and these organizations and these agencies that have been invaluable throughout this 2-year process of this project. First of all, thanks obviously to the High Desert Museum for giving me a venue to talk about this project. Big thanks to them. The second one, I want to thank the two organizations of the Native Fish Society and Raise River Dories. The Native Fish Society secured funding for this project. They were having an auction, and Raise River Dories donated the drift boat that the Native Fish Society auctioned off for the $10,000 that is funding this project. So thanks to both of those organizations. And I've also got so much advice and great leadership from many an agency and group. The U.S. Forest Service here in the Deschutes Basin, Nate Dochler, Jason Wilcox and their whole team, as well as the U.S. Fish and Wildlife Service here in the Deschutes Basin and ODF&W for sharing all of their knowledge about this basin and what they know about it, and have just been really supportive of the whole project. I also want to thank the U.S. Forest Service, the Rocky Mountain Research Station. We'll be talking about those guys quite a bit over in Missoula, Montana. They're the ones that analyze these samples. And Trout Unlimited, it was an organization that I'm highly involved with. They put a newsletter out to get volunteers to help me out with this project. So big thanks to them. And of course, the many, many volunteers that came out to help me take samples and to actually go out on their own to take samples. And that list is long. So I really appreciate all those guys. And Dr. Herzog brought out his class from OSU. This is an image of them. They took the samples at Trapper Creek above Odell Lake, just slogging through the snow, and they were troopers. So thanks to all of these people and these organizations for all of their help and support. So what we're going to talk about tonight is, first of all, we're going to talk about what bull trout are and why they're important. We're going to talk about this cutting edge DNA technology called environmental DNA, which has so many applications in so many arenas. And I'm just really excited about this technology. We'll talk about the project that I'm working on, and it's called the Range-Wide Bull Trout eDNA Project, sponsored by the Rocky Mountain Research Station over in Montana. And just, excuse me, a little bit on the challenges and the goals and the opportunities that I see for this project. So let's talk about bull trout. This is a pretty impressive picture. This picture of this bull trout is the world record bull trout, massive fish. This picture was taken back in I think the late 40s. Bull trout are actually not trout. They're a species of char, and they are related to the Dolly Varden and the brook trout. Actually, the bull trout and the Dolly Varden were considered the same species up until very recently. Dolly Varden are a separate species. Dolly Varden inhabit more coastal areas, and bull trout are more of an inland species. But bull trout are the only native char species in Oregon. So bull trout are characterized by having no teeth on the roof of their mouth, and they actually have light spots on a dark background on their backs, as opposed to a rainbow trout, a steelhead, or a salmon that has dark spots on a light background. So that's distinguishing for these species of char. Bull trout have very specific habitat requirements. They require incredibly cold water to thrive. And when I say cold, I mean in the upper 30s, the mid 40s, very, very cold water. They also require incredibly clean water, just like all of our salmonids. And then also very complex and connected habitat. Some of these bull trout are considered sort of resident, if you will. They don't wander too far from their natal streams. And then the other bull trout are actually quite migratory and will move from stream into a lake or reservoir or into the Columbia and up other rivers. Some travel up to 150 miles. So some bull trout resident, some bull trout migratory, and that's where that connected habitat comes into play. Conservation biologists are using bull trout as an indicator species for some of the effects of climate change, because these species, this bull trout requires such cold water. It's sort of the canary in the mine shaft for the effects of climate change, with warmer temperatures, warmer water temperatures, warmer air temperatures. So keeping an eye on bull trout and how they're doing. It's also an apex predator. You can see, especially by the size of this fish, it will eat anything it can get its mouth around: other fish, snakes, frogs, small mammals, ducklings. So play a really important part in the river ecosystem. Some of the threats that bull trout are facing, and they are considered a threatened species. They are ESA listed, so Endangered Species Act listed. They're considered threatened, and here are some of the threats that are most prominent for the bull trout. Climate change, we've already talked about the effects that those can have for all salmonid species and those warm temperatures. This particular char species, the bull trout, also requires very clean water. So some logging practices that put sediment into streams really do affect the health of the bull trout. Invasive species, also a big problem. What you're seeing here is, this is a brook trout that I caught in the Upper Little Deschutes. The brook trout is also a char species and inhabits the same areas as the bull trout and are known to actually hybridize with the bull trout. So problems there. Brook trout are not native to the West Coast. They are native to the Eastern United States. So an invasive species in this area. And lake trout, this monster was caught in Odell Lake and Trapper Creek have one of the two remaining populations in the Deschutes Basin, and lake trout eat bull trout. They compete for habitat, and just not a good species to have around a threatened, the threatened bull trout. And of course, I talked about how those bull trout want to travel. Some of them are quite migratory. So any fish barriers, whether that be a huge one on the Columbia or a smaller one more locally, are all threats to the continued existence of bull trout. Excuse me. So what you're looking at here is a distribution map, historic versus current distribution of bull trout. Bull trout are native to the Northwest, Washington, Oregon, Montana, and Idaho. You can see the red is where they used to be, the historical range. Now they inhabit less than 50%, less than half of their historical range. There used to be populations in California and in Nevada, and now they're relegated to quite often just the upper tributary streams. They do inhabit some lakes and reservoirs as well. Excuse me. So here we are in Bend, and I just wanted to point out, this is the Odell Lake population here, just southwest of Bend, and the Metolius population. If any of you are fishermen, fly fishermen, you know those famous bull trout that are in the Metolius River. And the reason that those fish are doing so great are because of changes in fishing regs, and because that river is just incredibly clean and cold. And that's allowed that bull trout population to really thrive locally. So according to the U.S. Fish and Wildlife Service, as well as ODF&W, bull trout are assumed to be absent in the Upper Deschutes Basin, except for those two populations that I mentioned, that Metolius stronghold, as well as the Odell Lake population in Trapper Creek, are the two that we know of in the Deschutes Basin. Other than that, it's assumed that they don't exist upstream of Big Falls on the Big Deschutes. So the project that I'm involved in is called the Range-Wide Bull Trout eDNA Project. That's a big mouthful. It is sponsored by the U.S. Forest Service over in Missoula, Montana, at the Rocky Mountain Research Station. And what you're looking at is a map of drainages throughout the West that the Forest Service is interested in and could be potential habitats for the bull trout. So as many of you know, the first step in the conservation of any species, threatened or endangered or otherwise, is to locate the existing populations. That's the first step. We have to know where they are. So what this project does is it hopes to establish the current distribution of bull trout throughout the West to inform conservation planning. So to get through that first step, they use two things. The idea is they use this climate shield model, which establishes the location of very cold water refugia, which could be natal habitats for bull trout. So they use GIS data, layers and layers, to determine where cold water refugia is, and they know that bull trout love cold water. And then the idea is to crowdsource collection of eDNA throughout these basins. So far, as of 2020, I guess it's 2022 now, they've collected a whole interagency, NGOs, lots of different groups that have collected samples and sent those eDNA samples off to Montana to get analyzed. And so far, they've collected over 15,000 eDNA samples since 2016. So really making a difference in those distribution maps and locating where bull trout actually are and where they are not. Just want to spend a couple of minutes talking about this technology. I just think it's really exciting to use cutting edge DNA technology in conservation biology. My degree is in molecular biology, and if somebody would have told me that I could combine it with the conservation of fish species when I was in college, I would have just been so excited. But this technology has only really come into play in the last 5 to 10 years, so it's pretty new. And what eDNA is, or environmental DNA, means that you collect an environmental sample rather than a DNA sample from a specific organism. So when I say environmental, that means that you could collect soil, water, which is what we're doing, hair that you might find left on a tree in the forest. You could collect snow that a species has stepped in. And just recently on NPR, they talked about how they have discovered DNA from the air, which has incredible implications. So it'll be interesting to see how that turns out. But you can collect eDNA for mammals, for fish, for amphibians, for any species. And what this technology does, and where its real strength is determining the presence of shy or rare and endangered species. Species that are difficult to find, or there's just so few of them, that this is the way to determine presence, is to just collect an environmental sample and determine whether or not they're in the area. Now, this technology has its advantages and disadvantages. I think the advantages are much stronger than the disadvantages, but we'll talk about both of those. One of the big advantages about this eDNA technology is that one sample can be analyzed from multiple species. So when I go out and I take a water sample and have it analyzed, I can have it analyzed for bull trout, and then I can also have it analyzed for rainbow trout and Oregon spotted frogs. I'm involved in Oregon spotted frog work and bullfrog work, so that's why that's in there. Up to 10 different species, one sample can be analyzed at this time for 10 different species, and they're working on making it 50 different species, all from one sample. And that's pretty powerful stuff right there. It's easier, cheaper, and less harmful than traditional techniques. When I talk about traditional, I'm talking about things like electrofishing, where an electrical current is put in the water and it stuns the fish, and then the fish are collected and they're counted and they're measured. And there's advantages to electrofishing. You can learn a lot from electrofishing, but if you're working with an endangered or threatened species, you want to minimize what's called take of that species. So sometimes when you think about take, you think about killing an individual member of that species, and actually take means a lot of things, but take also just means touching one of these animals, one of these fish, one of these endangered species. So by just taking a water sample or a soil sample, you eliminate that take, and therefore eliminate the need for federal and state permits that are needed for those takes. And those can be very timely to try to get, they can be expensive, and this technology removes that all out of the equation. And also very limited training is required to take these water samples. And when I say that, I get so excited about the implications of school kids, all kinds of groups that could get involved in this hardcore science project and conservation biology. And it's just really exciting to me, what we can do with this and who we can introduce this technology to. It does come with its disadvantages. You don't get any information on the life stage or the health or the abundance. For this project, we don't get any information on the abundance. Now, there are projects going on, eDNA projects. I'm thinking specifically of the lamprey project on the Lower Deschutes, where they're collecting eDNA samples, and their technique, they can actually establish a relative abundance of that species from their samples. But this particular project, we just get a yes or no. Is there bull trout? Is there not bull trout? There is, with DNA technology, there's always the potential for contamination, which is why we're very careful. We wear gloves. We're very specific on how we take samples. So we're very aware of that possibility and try to minimize it as much as possible. And then DNA can degrade depending on the conditions that it's in, the pH, the temperature, how much exposure to sunlight. All of those can degrade DNA and make it so that we can't detect the species that we're hoping to find. So definitely advantages and disadvantages, but again, just really exciting technology. I wanted to put this in here and I'll describe it. I'm going to play it a couple of times. This is a video, and I took this past summer. We talked about traditional techniques, and this is Forest Service personnel and ODF personnel that are in 40-degree water with dry suits, and they snorkel these streams, and they basically crawl up this stream to look for bull trout. And juvenile bull trout sort of hide in the daytime, so they have to go out at night, and they put on these dry suits and they climb into this incredibly cold water to find these bull trout. They're looking for other species as well. They're also, what I'm doing is I'm recording the data that they're seeing. I'm recording the number of fish and how big they are. So they do get more information than an eDNA technique. But I wanted to show this because it's just so impressive what some of these agency biologists do to help conserve this species. And they can really only be in the water for about an hour, and they just get too cold. Even though it's 90 degrees out in the middle of the summer in July, they just get too cold because this water is just above freezing. So just hardcore technique by these agency personnel to help establish how many bull trout are out there. And I was just really impressed with what they do. As far as my project, I don't get a dry suit on and get it in 40-degree water. I just stand on the side of the stream and take a water sample. And what you're seeing right here is all of the equipment that is required for an eDNA sampling excursion. So you're looking at the pump and the tubing. This is the filtering mechanism. So this is the first thing in the water. It's actually put in the water upstream of everything else. And there is this little tiny paper filter right here. And the water gets pumped through that filter and hopefully collects bull trout eDNA, and through the pump and into a bucket. And we actually collect the water that we pump to make sure that we're collecting 5 liters of water. That's just a control that the lab has set up to make sure that these sampling groups that are headed out are all collecting 5 liters of water, that the DNA on this filter represents 5 liters of water, and it's just a control for them. So we take a sample, we have gloves on, and we remove this paper filter with forceps, and we put it in a bag with desiccant, which is basically just a drying powder, and put all of the relevant data, where it was taken, dates and location, and we send it off to the lab to get analyzed. This equipment weighs about 40 pounds, which is quite a bit for one person to carry, especially when There are no trails that you're navigating overland and climbing over bushes and logs, and it gets quite heavy very fast. So this is what we take out into the field to collect a sample. And we also, I wanted to mention, we go out in the fall to collect samples. Some people can go out in the spring, some in the summer. And we've decided to go out into the fall for various reasons. One is the lack of mosquitoes, which is huge. And also bull trout spawn in the fall and are more likely to be in these streams that we're hoping to find them. So we go out in September and October to take these samples. I'm not going to spend too much time on this slide. I know it's a little busy, but I just wanted to show real quick what happens to these samples and how we get from taking a water sample in the Upper Little Deschutes to getting a yes or no from the lab in Montana. So we head out hoping to find bull trout water and bull trout DNA. We collect that sample in on that little filter, and we jet it off to Missoula, Montana to the lab. Excuse me. And they extract that DNA from that filter, and hopefully there's bull trout DNA there. The other thing they have at the lab is what's called a primer. And what this is a species-specific piece of DNA that is just specific to whatever species you're interested in. So we're interested in bull trout. This is a bull trout specific primer, which is a piece of DNA that sequence can only be found in bull trout. And this little star that I've put in here, that represents the way that they detect it, which is usually a fluorescence. They can detect the fluorescence on it. So what they do is they take that DNA and they hybridize it or bring it together with that bull trout primer. And they do something called PCR. If anybody is familiar with true crime shows or podcasts or whatever, you might have heard this term. A PCR is just taking a very small amount of DNA, even just one strand of bull trout DNA, and what's called amplifying it. And it goes through a series of heating and cooling with a specific enzyme, and it amplifies even one piece of DNA to get multiple pieces of DNA so that it can be detected and we can get to this yes or no answer. So that's a very general idea of what goes on in the lab. So I work in the Little Deschutes Basin. I am the Native Fish Society Steward for the Little Deschutes. And this is the Little Deschutes Basin right here that's outlined in red. So we're in Central Oregon. I'm in Sun River, talking to you from Sun River, Oregon. Everyone is familiar with the Big Deschutes. This is Crane Prairie here, Wiki up here. And the Little Deschutes is a tributary to the Big Deschutes. A lot of people don't even know that the Little Deschutes River even exists. And then Crescent Creek and Crescent Lake, all tributaries to the Little Deschutes Basin. And here are the headwaters here. So when you reach out to the Rocky Mountain Research Station and you say, I'm interested in taking bull trout eDNA samples in my basin, you pull up wherever your basin of interest is and you see this map. And this is of the Little Deschutes Basin. And then I've put a blown up version of it right here. So this is Crescent Lake. And these different color codes indicate what the climate shield model, that model that indicates whether or not bull trout might be in a drainage, that climate shield model has predicted the chances of finding bull trout in these different reaches of the Little Deschutes. So this is Whitefish Creek right here, tributary to the Crescent Lake. And according to the climate shield model, it has a 90 to 100% chance of finding bull trout in this drainage. So needless to say, this is one of the first places that I sampled 2 years ago was Whitefish Creek. We'll talk more about that in just a second. So this is what you would find if you're interested in getting involved in this project. Now, this is a pretty busy slide. I use navigating is incredibly important as part of this project. Quite often there are no trails where you go, so you have to know how to get to these sampling sites. I use a program called Onyx Hunt, and what you're looking at right here are pictures or waypoints or sampling sites that I have taken in the Little Deschutes Basin. So Lapine is up here, Highway 97 here, Highway 58, Crescent Lake, Odell Creek. So most of my sampling is taking place in the upper Little Deschutes. This is in the Big Deschutes here. This is Odell Lake and Trapper Creek. And just real quick, I want to mention this little part, this few sampling points over here. This is a place called Sellers Marsh and Round Marsh, and is a really special area that I'll talk a little bit more about, but it is also in the Little Deschutes Basin. So I'm just going to go through a series of these. Earlier, I talked about how there could be contamination with DNA technology. And so we're very careful about how we take samples. And one way that we do that is we take samples from the bottommost point all the way to the top. So we start taking samples down here at the bottom, and then we work our way up towards the upper reaches. And what that does is that minimizes contamination. If you can imagine going all the way to the top of this drainage and taking a sample, say there's bull trout here, and you walk down this basin and you've got bull trout on your boots, you've got bull trout on your hands somehow, you could then contaminate all of your next samples and get false positives. So you sample from the bottom up to the top. And each sampling site is about 1 kilometer apart as far as river distance. This is the Little Deschutes here, and then Clover Creek, which again is another spectacular place in the Little Deschutes Basin. And I'll show you some pictures of that. And again, we sample in the fall because that makes the most sense for us. And just an even closer view, once we get to the sampling site, we set up our equipment, we take an eDNA sample, and then we also note any other data that might be of importance. For instance, water temperatures. This picture was taken at the very top of Clover Creek, and the water temperature was 44 degrees, which is really, really a great temperature for bull trout. And I have a lot of hope for these samples on Clover Creek, which again is a tributary to the Little Deschutes. And we record lots of things that we see and lots of data because it might be important. It's important now, and some of it might be important in the future. So the next couple of images, the next couple of slides are images that I've taken, pictures that I've taken throughout the past 2 years that show what we see and how amazing these places are that we get a chance to visit. This is the Upper Little Deschutes River, and it's south. This part of the river is south of 58 and by the small community of Mowich. You can see the river is crystal clear. It is chock full of brook trout. If you're interested in brook trout fishing, this is a great place to go. But I will warn you, in June, July, and August, it is also chock full of mosquitoes, uninhabitable. So full of mosquitoes, but spectacularly beautiful wildflowers, meadows. So this is what the Little Deschutes looks like way up high. Not quite the headwater, it's just below that, but just a spectacular little river. Just some more images of what we find out there. I mentioned that we go out and take samples in the fall. You can see the fall colors here on Basin Creek. This is Sellers Marsh. I had mentioned that little space of a couple of samples off to the east of 97. Sellers Marsh is a high elevation marsh. It's really hard to get to. It's really remote, and it is a spectacular setting. There's a spring that comes out of the middle of it, and it was listed as 75% probability of finding bull trout. So I went up and sampled it and got negatives for Sellers Marsh. I'm going to mention this again because it's sort of a pet peeve for me. I just think this is a fascinating area and would love more attention on Sellers Marsh. So what you're seeing here, we go to these sites and we start at the bottom and we take samples and we work our way up further and further up the drainage. And what we have found in a lot of these drainages is just dry creeks. This is, again, Sellers Marsh right here, again, listed as 75% probability of finding bull trout. It is dry, as dry could be, and looks like it's been dry for a very long time. This is Whitefish Creek up above Crescent Creek, a 90% probability based on the climate shield model, and it's completely dry. And I'll talk about this again. So, excuse me, it's a little disheartening when you're headed out to collect samples and this is what you run into. But this is data that we collect. It's important for the lab to know that these areas are dry, and they note that. While we're out there, we also note other things that we see that aren't necessarily on our to-do list. For instance, this is a Cascades frog that I found in a spring, in the headwaters of a spring. The water was 39 degrees and this little frog was absolutely loving it. Just a gorgeous little frog. Bear scat in the fall. This was a very large canine print that I was not associated with any recent human prints, so who knows. But we talked about how you could take DNA from soil samples from many different things. And in theory, you could take a soil sample from this print and perhaps get eDNA out of it and know exactly what species made this footprint. And of course, you could find lots of things out about what's going on here with this bear scat. The other thing that we note is recent beaver activity. Beaver is sort of up and coming in really being recognized as critical for river habitat. And so when we see recent beaver activity, we note that too. That might be important in the future. Just a few more pictures of just some of the incredible things that we see while we're out there. Just incredible fall colors, that same little frog in that spring right there. Just beautiful. As far as the challenges of the project, there's a couple of challenges. I'm going to come back to this Whitefish Creek here. Again, this is Crescent Lake. This is Whitefish Creek. And again, according to that climate shield model, it had 90% chance of finding bull trout. So I hiked up there and I collected a sample 111-1 and I collected 11-2, and the remainder of this entire drainage was dry, completely and totally dry. So again, a little disheartening when you see that. But the other challenge that I was finding is access. There is a trail that goes along Whitefish Creek, but these samples over here, these drainages, there's no trail. And that's the case for most of these places. Access is very difficult. And quite often I'll only get 3 or 4 samples in a day because it's so hard to go overland and get to these sites. The access is just so difficult. And with the weight of that equipment, and I'm out there quite often by myself, it's limiting in the number of samples that I can take in a day. Again, we've decided to sample. We sort of discussed when the best time was to sample for bull trout. I've decided to go out in the fall. Bull trout spawn in the fall, so are most likely to be in these streams. And again, just less mosquitoes. I know it sounds basic, but it's important. And the cost of analysis and funding, as is the case with any of these projects, this is one of the big challenges is getting funding for these really important projects. So as far as the current status of this project that I'm doing, we collected 11 samples in 2020 from Sellers Marsh and Whitefish Creek, and we didn't get any positives. However, this past fall, we collected 43 samples that have yet to be analyzed. We did find 28 sites that were listed by the climate shield model as having a good chance of having bull trout that were completely dry. So again, we note that and send that off to the lab. And another reason I haven't had them analyzed yet is we're headed down to the Crooked River this week to collect more samples. You might know that the Opal Springs fish ladder was installed in 2019. Bull trout have not had access to the Crooked River for decades, and now we know that they're using this fish ladder and they're moving up into the Crooked River, which is so exciting. And we're going to head down there and we're going to take some eDNA samples and see how far up these bull trout have moved into a river that they haven't seen for decades. So anytime that fish passage can be established and you can see that fish are starting to use it, it's just really exciting. So the goal of this project was to establish remnant populations of bull trout in the Little Deschutes. We're still working on the answer to that question. If we do find some positives, the hope is that there will be special management and protections for those bull trout streams. One good note is that a lot of this potential bull trout habitat is already listed as critical habitat for the Oregon spotted frog. So it already has a high level, the U.S. Fish and Wildlife Service level of protection, which is great and a positive. And I already mentioned the opportunities, the community science engagement opportunities are just stellar with this project, with kids to get out there and maybe a STEM program. This is hardcore science that these kids could do and really make a difference. And I'm excited about that. And hopefully there'll be future funding for eDNA projects, not just for bull trout, but for other species as well. Again, I'm involved in the Oregon spotted frog conservation, and maybe we could start taking eDNA samples to establish presence of Oregon spotted frog. So as far as the future of where I see this project, there are really positive ongoing projects that the agencies and nonprofits are doing right now to help bull trout and all of our native trout. There's been a reintroduction of bull trout in the McKenzie River. There's constant removal of fish barriers. We need to keep working on that. Lots of habitat restoration that's going on in Subtle Lake and Link Creek. Trout Unlimited is involved in that for service. And then another big one is removal of invasives, which is a very difficult thing to do, but something that has to be done. As far as eDNA technology in the Deschutes Basin, the Forest Service in the area has really stepped into this technology and embraced it. And it looks like they're going to be using it to establish their distribution maps for different species, maybe detection of invasive species to really put resources towards removing those. So that's exciting. And again, being able to use this technology to determine presence of Oregon spotted frog habitat, that sounds really exciting to me. As far as for this project, the remaining of the funding is going to go towards sampling either the Big Deschutes, the Crooked, or in the Klamath basins. We've exhausted our possibilities in the Little Deschutes Basin. If we don't get a positive, then it's time to move on to a different place. But if we do get a positive, fingers crossed, we'll confirm that by eDNA analysis. We'll do another sample, and then the agencies would have to go out there and get a physical boots on the ground, eyes on the species to get a confirmation of the presence of bull trout if they truly are there. And just one of my last slides, what can you do? What can you do to help native fish? What can you do to help bull trout? The two that come to mind are these two great organizations, the Native Fish Society and Trout Unlimited. Both are doing spectacular work to preserve native fish and to conduct restoration projects, and they're always looking for volunteers to get involved. And I'll be taking more samples next fall. So feel free to reach out to me if you're interested in heading out to collect eDNA samples and get involved in this really exciting project. Here is my contact information. you can get on my website, threeriversenvironmental.com, or contact me through the email. And if you're interested in the eDNA project, more information, you can get on their website, and they have lots of great information on there. So any questions?
Thank you, Jodi. I know you can't hear us, but there's 57 of us watching you, and we really enjoyed that. So thank you very much. And there's also a really great conversation that's been happening on the chat. And I know you haven't been able to see that, but everyone is really enjoying your presentation and this information. So I do have a few questions. One is from Nate, and he asks, any chance for contamination of a water body from other wildlife, like waterfowl that might fly from one stream to another?
You mean if you were sampling for some sort of species of duck? Is that what I'm
it sounds like it could be a species, like, yeah, waterfowl, or potentially even, yeah, bull trout. Let's say they have water on them and they've flown from one location to another on their body. Could that have contaminated the next stream?
Well, I don't fully know the answer to that question. That sounds like it would be a pretty dilute DNA sample. Again, this DNA technology is very powerful, and even a few strands of DNA could be picked up by this PCR reaction and amplified, and you could get a false positive. That would be a pretty dilute sample, though. The chances of collecting that, unless you're right under the bird, right when it lands, I'd have a hard time believing that it would contaminate your sample. But I like that that's how they're thinking, and that's how careful that you have to be with this sampling, because the PCR reaction is so powerful. It could take very small parts of DNA and amplify it. So great question.
Thanks. We did get a question from Cindy, and someone also answered that. Looks like Stevie answered it, but you could maybe comment on this. What are the current catch and release regulations for bull trout?
Oh, well, that depends on where you are. You can fish for bull trout in the Little River. It's a catch and release. That it's my understanding. I will say that I don't fish for bull trout, so haven't really educated myself on where I can catch them or try to catch them. It's my understanding that you can fish for bull trout in the Metolius because that population is thriving and it's healthy. You have to release them, but you can fish for them. Other places you will get bycatch. You can't target bull trout in most places because their numbers are so low. I can't speak to the regs in Montana and Idaho. All I can speak to are the regs in Oregon, and the Metolius is one of the few places where you can actually target them. Otherwise, it's definitely a catch and release unharmed.
Mm-hmm. Thank you. Another question is there a similar technology, this is from Dana, our executive director, is there a similar technology to some of the salmon forensic work that researchers also work on?
Could you read that one more time for me?
Yeah, sorry. Is this a similar technology to some of the salmon forensic work that researchers work on? I don't know if you know anything about salmon forensic work, but
I don't know anything about salmon. I don't know anything about salmon forensics. I know that this technology is deployed in lots of different places. They use it to determine whether or not somebody is bringing a rare and endangered species over the border. They can take samples and determine if, what species it is. Yeah, I don't know anything about salmon forensics and what it's being used for. Sorry.
No, it's interesting. Yeah, potential connection there. Another question from Stevie is, how far up the Deschutes do bulls go above Lake Billy Chinook up to Steelhead Falls, ?
According to the critical habitat maps established by U.S. Fish and Wildlife Service, and I should have mentioned, most of us that know about fish or associate with fish, we think of National Marine Fisheries as sort of the agency, the lead agency for most of our fish species. And the bull trout are a little bit of an oddball in that the lead agency is the U.S. Fish and Wildlife Service, which, because this is more of an inland species. So the U.S. Fish and Wildlife establishes critical habitat maps for all of the species under its jurisdiction. And it's my understanding that on the Big Deschutes, that critical habitat ends at Big Falls. So it is believed that there are no bull trout above that, save for the Odell Lake population, the Trapper Creek population, and then, you know, obviously on Lake Billy Chinook on the Metolius, but that's below Big Falls.
Okay. Crystal asks, maybe she missed it, but was there evidence of bull trout in Trapper Creek or Odell Creek?
So the Forest Service and ODF&W and U.S. Fish and Wildlife know that there are bull trout in Trapper Creek. So we actually went and sampled Trapper Creek because they're not sure how far up the bull trout are in that particular drainage. So that was actually sort of a different thing. We actually started at the top of that drainage and worked our way down because we knew that bull trout are in Odell Lake and in the lower reaches of Trapper Creek due to the snorkeling surveys that those agencies have conducted. However, those snorkeling surveys are limited to those lower portions of the river. And so what we did is we hiked all the way up and took samples on our way down. We haven't had any of those samples analyzed yet, so we don't know how far up Trapper Creek the bull trout are, but the agencies know that they are in Trapper Creek, but how far up is still to be established.
Thank you. One question from Molly is, what got you so stoked on fish? How did this work start to pique your interest?
That's my sister asking that question. I've been a fly fisherman for 20 years, and I think it's the natural flow for fly fishermen to go from fishing into conservation. I've just seen it happen so many times with so many people in Trout Unlimited and Native Fish Society that as a fly fisherman, it's just natural to want to conserve what it is that you hold so dear. Again, I have a degree in molecular biology, and the idea of conservation biology and helping these native fish, along with getting to do molecular biology, which I just think is the coolest technology out there. It's a dream project for me. I don't get tired of talking about it to anybody that wants to listen.
Thank you. And I will say that there's a conversation going on in the chat of just a lot of people discussing where do bull trout exist. And Mike messaged some of the hosts and panelists at the beginning saying that there is a population of bull trout in the upper middle fork of the Willamette River as well. But yeah, anyone can read through those comments, I think, and see some of that conversation. And we also got a few questions about like how people can contact you or potentially volunteer. So we'll definitely keep your information there up on the screen so people can email you. And that sounds like the best way to get a hold of you. Here's another question. Oh, go ahead, Jodi.
Oh, I just wanted to mention, I can't remember if I mentioned it or not, but there has been a successful, what I believe is a successful reintroduction of bull trout into the Mackenzie River. They took fish from the Metolius and introduced them into the Mackenzie. So reintroduction efforts are out there, and some of them are successful, which is great to hear.
Thank you. One question is, do bull trout need a lake in the river system?
I don't believe they do. They're not like, and I'm sure that there's biologists that have attended this that know more than me about the life history of bull trout, but they're not like kokanee. They're not like sockeye salmon in that it's required that they spend 2 or 3 years in a lake before heading out to the ocean. It's my understanding, and that they could survive in just a stream if there isn't a lake available. Again, many of them are migratory, and they head down these rivers and into lakes and up into other rivers, and they do hang out in lakes and reservoirs. But it's my understanding that a lake is not required. So if there's a biologist on here that knows otherwise, I'd be happy to learn that.
I'll let you know if
Perfect.
We have a couple minutes. If anyone else has another question, go ahead and type it in and I will keep an eye out for that. One question I actually have. Go ahead, Jodi.
Oh, I just wanted to mention before people started logging off, I had mentioned I'm involved in Oregon spotted frog work, and I'm involved in bullfrog control, which bullfrogs are a predator of the Oregon spotted frog. So just speaking of volunteering, if going out at 9 o'clock at night on a summer evening to find bullfrogs sounds like a good time to you, then reach out to me as well. It's not just native fish, it's native frogs too that I'm really enthusiastic about.
Thank you. Yeah, we did have one question of someone who asked if we could send your contact information out to everyone who had RSVP'd for the talk today. So I think we could probably make that happen. Sounds like you've got a lot of volunteers here tonight. So
excellent, excellent.
Well, that actually puts us right at about time, and this is really wonderful. So thank you again, Jodi. This was fantastic. This is being recorded, like I said, so it will go up on YouTube here in the next few days. And we all appreciate this information. This is what I love about Natural History Pups, is that we get cutting edge science delivered directly to the public. So thank you very much. Goodbye,
Perfect.
everybody.
Thanks, Haley. Thanks to the High Desert Museum. Have a good night.
Bye-bye.